In the UK, Rutland-water spans over 1,000 acres. As Rutland-water is a triple SI site, this makes it one of the best nature reserves in the UK due to its huge diversity of freshwater species. One of the most abundant is the class Insecta, which throughout their 400 million years of evolutionary history have adapted to both lentic and lotic freshwater habitats. Utilising our selected breeding strategies such as high fecundity rates and seasonal generations to maintain their genetic diversity in these isolated pockets of freshwater environment. However, poor quality water sources will be unsuitable for these animals to be able to reproduce, containing high levels of pollutants such as farmland runoff, sewage, heavy metals, and pharmaceuticals. The effects of these pollutants include a lower oxygen level in the water, an increase in the level of eutrophication, leading to excessive plant growth, a reduction in the overall fitness of the individuals living there, and a reduced fecundity rate of these insects. Now, for sites like Rutland-water, these bioindicator species can be assigned a BWMP score in order to determine water quality. For example, mayflies, Ephemeroptera, and stoneflies, Plecoptera, are highly susceptible to pollutants during their larval stages, meaning that water sources that can support their life cycle score a 10 on the BWMP score. Species such as diving beetles, Dytiscidae, and water boatmen, Corixidae, can survive different pollutant levels dependent on species, some being more sensitive to water pollution and some being much hardier, meaning these water sources score a mid-range 5. And species such as midges, Corixidae, and worms, Olingochiata, are super hardy and able to survive much higher levels of pollutants in water sources, meaning that they score much lower on the BWMP. During this assessment, a more in-depth approach was taken, utilising biological indices on data collected on one family in particular, and that family was Corixidae, as these benthic invertebrates score a 5 on BWMP. This means that Corixidae can survive in low abundance in poorer quality water sources and show higher abundance and species variation in higher quality water. In order to collect these Corixidae for further analysis, aquatic sweep netting was used on each site surveyed. For each site, 5 sweep netting efforts were performed for 3 minutes each in order to get consistent collection of Corixidae per site surveyed. The sites that were used at Rutland-water for this survey were Lagoon 3, which contains chemicals from chlorination and pharmaceutical products as a byproduct of water treatment from the Anglian Water Treatment Facility. The main reservoir, which receives outfall of treated water from Lagoon 3, and Lagoon 5, which is an isolated lagoon that receives minor amounts of agricultural runoff pollution, such as low levels of nitrates and phosphates. Now, from this sample, the males were able to be identified due to the ease of their identification, meaning that with a useful key, such as the Savage key on water boatmen, it increased the efficiency of the study. The species data collected on the Corixidae from the sample taken to the lab are then converted into biological indices in order to detect the water suitability at each site, as well as species variability. This surveying technique is actually preferred by nature reserves such as Rutland-water, as instead of using a BWMP and getting a general overview of species availability, as well as your water quality data, by using biological indices to break down one family in particular, you're able to get very specific species data, whilst also being able to detect water quality. Now, if we were to take a look at these two cluster diagrams taken for the raw data of each site surveyed, in terms of genus and species at each site, Lagoon 5 and the main reservoir on both of these showed a much higher abundance of individuals. If we were to look at the genus frequency, we can clearly see that Lagoon 5 has 3 different genus on their site, whereas with the main reservoir and Lagoon 3, they have the same proportion of genus to one another. Using this box plot here, we're also able to see the rarity and the presence of each species at each site in comparison to the number of species that appeared. As we can see from this, Distincta was highlighted as a clear rarity amongst the sample, whereas Phileni showed up to be one of the most common. However, from these graphs of the raw data, it's clear to see that Lagoon 5 had the most species diversity as well as genus diversity in comparison to the other sites. The biological indices used for this assessment included the Shannon Index, the Evenness Index, the Burger-Parker Index, and the Simpsons Index. Now, if we take a look at the values collected from our Shannon Index, you can clearly see that Lagoon 3 and the main reservoir had significantly higher figures than Lagoon 5. Due to the Shannon Index bias towards species in rarity, we can understand why Lagoon 3 received such a high result from this indices due to its reduced population. But why would the main reservoir receive such a higher result than Lagoon 5? This could be due to the main reservoir housing species such as Distincta and Wollastonii in smaller proportional abundances than Lagoon 5, highlighting this site to have species in rarity regardless of the number of individuals. The Evenness Index found Lagoon 3 to have the highest result, whereas the main reservoir had the lowest, which is justifiable due to Lagoon 3 having such a small population. Evenness works by using the relative abundance of species at a site and comparing it to the number of individual species. Now, considering relative abundance is involved, this means that species richness does not always mean species evenness. This means that if a site has a larger number of species, sometimes this can reduce the result of the Evenness Index as the relative abundance of that site has increased. The Burger-Parker Index found Lagoon 3 to have the highest result due to its higher proportion of common species such as Phileni, and it also found Lagoon 5 to have the lowest result due to that abundance of common species being diluted by the rest of the species variability. This indices is considered one of the more reliable of the indices due to the lack of bias and its ability to use the abundance of common species as a ratio to identify where the common species mostly occur. This means sites with more rare species, such as Lagoon 5, will have a lower result on the Burger-Parker Index. The Simpsons Index shows a lower value for multi-species communities, or the probability that two species picked out from one population will be the same. This subsequently creates a natural bias towards more common species. As Lagoon 3 had proportionally more common species, the results were much higher, whereas for sites with a little bit more species variation, such as Lagoon 5 and the main reservoir, that result was again diluted and reduced. So, what can we actually conclude so far? Well, it's fair to say that Lagoon 3 has a pretty even population of more common individuals, which are likely to be more hardy to the pollutants coming from the Anglian water treatment facility and dumped straight into that Lagoon 3. The main reservoir has a pretty good range of species variability. However, the evenness of these species is slightly off, with some of the more rarer species being in lower populations than the common species. Now, this could be to do with the outfall from Lagoon 3. This outfall could possibly be polluting even bits of the main reservoir, meaning that rarer species can struggle to maintain their population. Lagoon 5, however, houses a vast range of species variability, all with pretty reasonable levels of evenness, which shows the benefits of this lagoon being isolated from Lagoon 3 and not receiving that treated water from the Anglian water treatment facility, despite this site still receiving pollutants from agricultural runoff. In conclusion, the treated water received by Lagoon 3 from the Anglian Water Treatment Facility has a clear effect on species variation and abundance at this site, also possibly affecting surrounding water bodies such as the main reservoir from the outfall of water from Lagoon 3. However, to ensure this result is correct, this test would need to be repeated a number of times to ensure the results between different tests are consistent. But from what we can conclude right now, to ensure that Rutland-water maintains its biodiversity of freshwater invertebrates, these isolated lagoons such as Lagoon 5 need to have regular management and maintenance to ensure these species remain at that site. For further reading on this topic of freshwater ecology and biology, feel free to refer to these citations down below